Last lecture, we covered some basics of pharmacology. So that's kind of how drugs work, how they're processed by the body and that kind of thing. And in this case, now we're looking at clinical trials and what we call the placebo effect. And again, this kind of background information is really important to understand how we actually come up with new medicines and how we judge or decide if things are actually effective as medicines and what the process of that is currently. And this is all going to become really relevant background for understanding things like medical cannabis, which is one of the main kind of topics that will come up later in the course. So we need to understand kind of what are the kind of rules and regulations and the kind of questions we're asking when we do clinical trials or test a new pharmaceutical, for example. So the main kind of purpose of doing clinical trials or clinical studies of new compounds, new medicines, is to provide evidence to see if they have safety and efficacy. So efficacy is another word for do they work? Are they efficacious? And it's also a way of validating the science. So you're bringing scientific validity and integrity to the process. They are expensive, very expensive to conduct clinical trials. And they are required for regulatory approval for various different regulatory bodies so for example, the FDA in the US and similar scenarios in Canada. So these are required if you're going to actually call something a medicine and prescribe it to patients for a specific indication, like pain, for example. So I've got a number of different kind of diagrams or visuals, just that often they're saying kind of the same thing, but they're just to help us. I always find visual things quite a good way of getting understanding a process. So the first thing that happens is there's lots of what we call basic science, and that's the kind of research I do in my lab. So we're looking at things in models of, say, for example, of cells or tissues taken from a different disease or something like that. So we're looking in the lab setting to try and figure out if we can find a new medicine. And then preclinical testing as well as kind of different types of testing that will take place before that compound actually gets to clinical trials. Now, clinical trials, the kind of broad definition is that this is happening in people, so that the compound's then starting to be tested in people. Now, this particular diagram has approximately 9 years here for that timing. Now, that will vary a lot depending on the medication and the situation. So this is just a kind of example, and different drugs for different illnesses can take different lengths of time to go through this process. And as we know, for example, the vaccines for COVID was really shortened for various reasons. And in other instances, this clinical process can be a lot shorter depending on the patient population and what the different illnesses. For example, if a medication is being tested in a population of people for whom there's no other kind of hope of treatment, so it's a terminal illness and they've tried all other things that could be tried, then there may be ways in which the clinical trials process might be speeded up. But these are all additional bits of information you don't really need to know for this course. It's just understanding the framework. And then after that process is complete, then there would be what's called approval. So the drug's then approved as a new medicine or a new drug for treating whatever it's been tested in. And it's important to note, this is kind of obvious, but not always obvious when it comes to medical cannabis and things like that, that this compound has been tested it's been approved for a specific illness. It's not just being approved to use in anything. So the clinical trials process would involve testing this molecule in the patient group for the illness that it's going to be approved for. So you wouldn't get approval for a drug for pain and then say, oh, it's approved, we're going to use it in anxiety. Now, sometimes those things can happen. It's called off-label, but in general, what you've got is evidence here of safety and efficacy in a particular group of patients with the illness. But you also have in phase 1 of the trials, you have various phases of the trials, which we'll talk about. And in phase 1, you have testing in healthy individuals, the first phase. So you do know that this compound from these safety trials initially is safe, in that it has been tested in healthy people and looked at for over a shorter term for acute toxicity or something like that. So that is included in the trial. So the basis, the different reasons for doing this, and what does the approval mean? So it means that the target condition, so you've analysed like the target condition and available treatment. So the target condition is what the illness that you're trying to treat. And the reviewers would be analysing this condition or illness for which the drug is intended. And importantly, they're looking at the landscape, like, do we need another medicine? Is there a really, really good medicine that's not too expensive and has great results and few side effects? In which case you'd be thinking, well, really, is this new medication any better? Does it have fewer risks or greater benefits? Back to that risk to benefit thing that I talked about yesterday. So, for example, a drug intended to treat patients with a life-threatening disease for which there are no other therapy may be considered to have benefits that outweigh the risks that would be unacceptable for another condition. So that's a key point there. So they're looking at the illness and the whole picture. And that again is important in terms of when we start to think about medical cannabis. So they're then looking at the benefits and risks. Again, they're looking at all this information that's submitted after the clinical trials happen, which I'll tell you about how they do the clinical trials. So taking it, there might be some uncertainties or whatever. So they're taking into account all this information and trying to make some kind of judgments from that. And so they're really looking for evidence that the drug will benefit this target population with this illness, and that the benefit will outweigh risks and uncertainties as well, because there might be unknowns that remain. And then also strategies for managing risk. So all drugs have risks, and that includes the herbal type things, as we've talked about already. So nothing really is without its risks. Often that can depend on things. It can depend on a whole range of variables, including dose, for example, at-risk populations, different vulnerabilities in different groups of people. It can depend on the nature of the illness or the mild or severe nature of the illness and of the side effects. So there's a whole bunch of things. And then there can be a risk management strategy to manage the risks associated with that potential medicine, so that people who might be at specific risk from a side effect could be excluded from being prescribed that compound. And that does happen. Or the label could include specific warnings, for example, things that might happen with that particular medication. So these are the kind of frameworks that are in place for clinical trials. Now, of course, the clinical trial process and the whole thing is not without problems. We don't really have time to go into all of that in this course. We'll mention some of these, but there are all sorts of ways in which this hasn't always worked perfectly, and which there are still kind of some criticisms made of the process of approvals and so on. But this is the current situation in our context in terms of approval for a medicine for a specific condition. So in terms of the different stages, again, of coming up with a new medicine, so you've got an initial drug research. Researchers are often screening many molecules. So for example, in my lab, sometimes we'd be looking at maybe hundreds or thousands of compounds, and you're looking at them in cells, and you've got your target, say, receptor, say that could be CB1, and you're trying to look at molecules that might actually have an effect on CB1, and you're gathering preliminary data on whether it works and it's specific. Of course, things like AI and computer-aided drug design and so on have speeded up some of the process, and sometimes people are now looking at virtually at millions and billions even of structures to see if they might fit into that, to be a key for that particular lock of that drug target. So that's the initial research. And then preclinical studies are done in, for example, in rodents, so maybe in mice or in cells, looking for serious levels of toxicity, for example, heart effects or liver effects, or also teratogenic effects. So that's effects on, so if someone took the drug while they were pregnant, what could be the effect on the fetus? Those kind of things led to really major, like terrible disasters in the past, where drugs had effects and people were taking them, say, for morning sickness and didn't know that there was effects on the baby. And so now some of those things are now built into preclinical testing, so there are certain things that have to be tested before a compound would ever be allowed to be in a human for phase 1 of the trials. So there's a number of things included in that. But even then, some things have happened that have been kind of that have potentially gone wrong. But it's much improved over the way it used to be prior to the more of a framework that was more better design of a clear steps for testing preclinically. So then it's the clinical trials. So all drugs marketed or sold in Canada must have been studied in clinical trials, and the information gathered is then presented to the Health Products and Food Branch of Health Canada, and then they will make this judgment of whether the drug becomes approved for this illness. And they're gathering all this different information, including dosage. And it's important to note that here. I'll be talking a lot, it will come up over the course of this, different aspects of this course, about things to do with the dose of cannabis and the amount that's being taken, and the different ways in which that can be helpful or problematic. And dose is a really important thing in pharmacology. It's a really important thing in toxicology, and it's a really important thing in terms of the way clinical trials are designed. So when a clinical trial is designed, they're actually figuring out, do we give this new medicine, is it going to be given to the person once a day at 10 milligrams? or twice a day at 20 milligrams. And it's all to do with things like the pharmacokinetics of the molecule and how effective it is. And there's no way that you would suddenly that all the clinical work would have been done, saying that 10 milligrams a day is helpful for pain or sleep, and then your family doctor would suddenly say, oh, but I'm going to give you 100 milligrams, I'm going to multiply the dose and it'll be fine. So that's kind of, again, a framework for how important it is to understand how much of something is being taken and what the implications of that can be, and how a lot of that is addressed with pharmaceuticals in terms of the dose, and isn't always addressed when things are more like a natural product, where this kind of rigorous analysis of the safety of different doses and amounts has not perhaps been analysed or understood to the same extent. And that's one of the unknowns that's really important for us to figure out as we go forward with cannabis. So often the results of clinical trials were then either finding out if the drug is effective against a placebo, which we'll talk about, which is just a kind of sugar pill, it doesn't contain any of the drug, or whether it's being compared against an existing drug that's used for that illness already. Because sometimes it's important to say, well, is it actually better than that one, or is it not? Is it worse? In which case there's no real merit in approving that new compound. And then there's the submission. They submit this document, new drug submission, and then that would all be analysed with all the different things I've said to reach a decision of whether it's approved or not. So that's a big, long, expensive process to go through. And again, this is just another visual, which is another representation of this, which kind of helps just represent what I've said. So you've got the preclinical testing, then the different phases of clinical trials, and then you've got data analysis, and then there's follow-up of continuing to monitor the drug after it's approved, which is a really important part of the process, monitoring whether other things emerge after it's approved, because it can take years for certain problems to emerge with a pharmaceutical. And you may know of examples of that, where certain subgroups of people have suddenly been shown to be at risk, or there's a risk associated with a certain medication that only shows up many, many years later when they retrospectively analyse groups of people who were taking it or weren't taking it. And they find that the initial clinical trials were conducted over maybe 3 to 5 years. But if someone's on the medication for 10, 20 years, there are other problems or not. So that monitoring is really important to do. And that has yielded all sorts of different results and still is. So what the phases of the clinical trials, you've got the initial dose findings. So you're trying to figure out what dose might be good for a clinical trial, and then you're proving the concept. You're in the first trial, you're also looking for initial warning signs or red flags in these healthy volunteers. Initial signs of toxicity, monitoring all sorts of different things when people take. So you start with a very low dose, keep monitoring, increase the dose, and look at the levels of the drug in the plasma and the blood. And then initial proving of the concept. So in a small group of people with the illness, you're actually you're doing a comparison. Does the drug show any evidence of being effective? And then you're doing a larger study, which may involve a lot more patients in multiple locations around the world. And that may be comparing with a standard therapy, or it might still be with a placebo, but it might be with a standard therapy that's currently being used for that condition. But for some conditions, there is no therapy standard. So it might be compared with a placebo, which is, as I say, doesn't contain any active drug. So again, this is a summary of the phases of clinical trials. Healthy volunteers, trying the drug for the first time, you're looking at the initial PK, pharmacokinetics and pharmacological effects. Look for adverse reactions or unexpected things. Then you're looking at the effectiveness phase, a larger group of individuals with the pathology, so with the illness, like pain, for example, and you get this initial evidence of whether it's actually safe and has any effectiveness. And then that would justify spending much more money on doing this big phase 3 trial, larger groups of people, multiple sites around the world, to confirm the effectiveness, monitor side effects, and compare with commonly used treatments or with a placebo, and then analyze that information as well. And then there's the monitoring phase, as I said, which is very important in terms of these long-term effects. And that's where, again, this is really relevant to medical cannabis or cannabis use, because there's all sorts of medical use of cannabis, whether it be prescribed or not, or whether it be self-medicating. And the questions are, are we really monitoring these longer-term effects? Because it's not been often it's not been part of this kind of clinical trial process, and therefore there's not necessarily this rigorous or monitoring that should be happening of these long-term effects. Now, we could also say that doesn't always happen so effectively with pharmaceuticals either, and some of these long-term effects can suddenly start to appear or emerge, and so it's there's always things that we're trying to figure out how things could be done better or differently. And again, this is an example from a particular illness called FOP, which is a particular bone-related disorder. But this was something that was kind of an infographic visual that kind of shows again the approximate length of time. And you can see in this particular condition, it shows you the time for each of the phases, and this will vary for different conditions, as I mentioned, depending how long it takes to figure out if something's happening. So for some conditions that you really need a long time to see if there's any improvement happening. And then in this case, it shows you the number of people who were involved for this trial. This is a rare disease, and so there's much smaller numbers of people here in this one because it is a rare disease. And so it's hard to find large numbers of people to conduct the trials. But in other non-rare diseases like chronic pain, there could be hundreds and thousands of people involved in these trials. But it shows you here what the purpose of each one is as well, which I've mentioned, which is helpful to know as well. And it also shows you the drugs that move from one phase to another. So, there's an attrition rate, there's a loss of compounds or drugs as we go through those phases. So lots of compounds might be tried in phase 1 and then found that actually there's some red flags in the healthy individuals, or the dosing isn't working, or it's not getting into the plasma properly, or and then phase 2, you might do phase 2 and find it doesn't work. And so it never gets as far as a phase 3, but you've still spent a huge amount of money on the preclinical and on the phase 1 and phase 2. So there's a lot of failed drug trials, and that's because of just the nature of the challenge of coming up with new treatments for complex, difficult conditions in people, which often have a lot more variables than the preclinical tests that have been done. And this shows you again, it's an example of the preclinical. People are testing in the lab tens of thousands of compounds preclinically in a lab like mine, and as I mentioned, with virtual screening and AI, now there'll be millions of structures being tested. But that funnel gets smaller and smaller until you eventually end up with one compound, hopefully making it through all these hurdles of potential, coming up with a molecule that hits the target, coming up with one that actually doesn't cause teratogenicity or affects heart valves or liver toxicity or any other carcinogenic effects, then getting through phase 1, phase 2, does it actually work, phase 3, and multiple all around the world global testing, and then getting through the review and then getting it. So you start to realize the enormous undertaking of a clinical trial and how expensive and time consuming that actually is. But in some ways, you can see that there's many aspects of the quality of the data and the information you're getting that would be severely lacking if you hadn't done any of this before. And this is showing you some of the costs of clinical trial costs. So you've got here, and it depends on the illness. So different illnesses, it's more expensive. So pain and anesthesia, for example, you've got $100 million to get to phase 4, which is this monitoring effect. So and you've spent before even by getting to phase 2, where you might find it doesn't work, you've spent $20 million. And then you've gone through phase 3, 60 million, and it may then not work. So that's the kind of cost you're looking at, and it varies from condition to condition. So in that context, you can imagine why there's sometimes a reticence to conduct clinical trials for things that people might be taking anyway, like or self-medicating with. So if people are already using something that's kind of herbal or plant-based, and the companies that are selling that already, accumulating large income from those sales, then the question is, who's going to fund the clinical trials, and are people willing to spend tens, if not hundreds of millions of dollars on a placebo-controlled clinical trial on the particular product? So this just puts a context around some of the things we'll be talking about as we go on with the course. So clinical trial registration is an important thing that, it would seem logical that all clinical trials should be registered and reported, but that wasn't always the case. And in 2017, which actually isn't that long ago, the World Health Organization, some of the largest funders and international organizations agreed on new standards that require all clinical trials that they fund and support to be registered and the results to be disclosed publicly. So it's only in 2017 that this particular group, the WHO actually came up with this. And at this point, currently, about 50% of clinical trials go unreported, and that was in 2017, often because the results are negative. So what was happening was that people were conducting clinical trials and they weren't reporting the fact that the compound didn't work. Partly because of their reputational damage for the company, but also that this could also even be applying to things in the realm of medical use of cannabis. So if something works, that's a great big PR campaign that it's worked, and if it doesn't work, the clinical trials were not necessarily reported. And so that's really a problem in terms of transparency and understanding what information there, and of course this applied to pharmaceuticals companies hugely as well. So that means that it leaves an incomplete and misleading picture of the risks of benefits of a whole bunch of things: vaccines, drugs, medical devices, that could lead to harmful results. So people have been really trying to tackle this, to improve this framework and make sure that it's actual legally binding reasons why people have to report these things. So in Canada in 2016, this was an article from CBC, why we've been deceived, many clinical trials are never published. And so really, again, this was happening relatively recently, that trials weren't being published, and those were usually the ones where it was found the drug didn't work or even had side effects that again weren't necessarily being reported. But NCIHR, the Canadian Institute of Health Research, which funds a lot of these trials, has now signed up to this World Health Organization's joint statement, and so there's now a requirement and legal requirement for actually reporting clinical trials outcomes. And so this is just anyone who gets funding from this research organization to conduct a trial then has to register the trial and then has to make the summary of the results publicly available within 12 months of the last visit of the last participant. So if they're conducting a clinical trial, the last patient completes the trial within 12 months, they're actually required to publish a summary of those results. So that would include obviously negative results, so where things hadn't worked. And the FDA in the US has made this mandatory trial registration, and later results reporting were proposed to mitigate this selective clinical trial reporting. And there's this FDAA was enacted by the Congress in 2007, requiring registration of all non-phase 1 clinical trials involving the FDA regulated interventions and reporting on that as well. So different places around the world have been different timescales in terms of doing this, and there's been different levels of compliance with these legal requirements as well. And so this was this is actually a relatively recent paper looking at compliance with the legal requirements. And you can see who sponsored the research: non-industry, industry, and the US government, industry collaborator, a US government collaborator. And so you can see they've got trials due to report, trials with any results, and compliant trials. So here you can see that the total number of compliant trials, so that's compliant with reporting the data within that time, you've only got 41% there that are actually compliant. So there's still this problem with people not necessarily being compliant with this requirement to report all the data within a certain length of time. And so and it shows you, depending on the funder, the different breakdown of that. So this is still something that's being monitored and looked at as to whether the reporting of clinical trial data is actually whether people are compliant with this or not. And the other paper that's quite interesting on this is this shows you is worth really kind of pondering on the cumulative effect of reporting and citation biases on the apparent efficacy of treatments. And this was in the case of depression. So you've got negative trials, this wee red dot here, positive trial, SPIN. Now, SPIN is in terms of how the results been reported in the media and by the company. Mild spin, so they've obviously found a way of distinguishing. They've got a definition of this paper of spin and mild spin. So whether mild spin is just a one press release, and major spin is like huge splashes all over major media, and then no abstract. So there's no reporting there. So you can see here that as you go across these different scenarios, you've got displays the initial complete cohort of trials. So this is everything that's been done, and you can see there in this case there's a kind of about 50-50 of positive and negative trials, so ones where the drugs have worked and ones where they haven't worked. And then while B through E show cumulative effect of different biases. So you can see as the biases start to get introduced, the impact of the negative trials seem to suddenly drop off, whereas the impact of the positive trials are actually starting to be inflated. So you've now got more focus on the ones that worked and less focus on the ones that didn't. And so you've got then suddenly this, whether it be a perception of the people prescribing, the MDs, whether it's a perception of the patients or relatives or just everybody in general, have got this perception of and this knowledge of the positive trials. And suddenly the ones that didn't work have then faded into the background. And this is really super interesting in terms of how things work with the information that we get. And that's why it's really important that we find tools to critically evaluate the information we have, critically evaluating who's telling us what and what kind of conflict of interest they have in giving us the information we're getting. And what does the data really say on the effectiveness and safety of something as a medicine? Now that this initial 1A here, there are lots of positive trials, like the drugs did work in a number of trials, and so that doesn't mean that the negative ones would mean you shouldn't use it, but it's just being able to actually come up with a well-informed knowledge of, and something like cannabis, for example, it's legal, it's widely available, it's widely used, and sometimes it's being used as a medicine, sometimes not, and sometimes it's being used as a medicine with self-medicating, so not necessarily being prescribed by a physician. And so the question is, what's the status of this A for negative versus positive trials? What's the influence of potential spin, be it big spin or small spin, and what's happening to kind of affect our overall perception and what we feel is the reality versus what the studies are saying. And this is the world we live in with everything now. It's not just in terms of medical treatments, in terms of so many things. And of course, the A there may have flaws in it too, as we've talked about. There could be flaws in the initial data that you're trying to build your solid information on. But anyway, I thought that this is very thought-provoking kind of study, and one that we can kind of think about again as we're discussing the topic as we go through the course. So randomized controlled trials. So just a little bit more on how these randomized controlled trials work. Why is it called a randomized control trial? And they're also called double-blind placebo-controlled trials. So you've got the study population, and they are then randomly assigned to get drug or placebo or current treatment. You've got outcome measures and then various things that you measure. Clinical outcomes, so does the illness get better? Biomarkers, various biomarkers, say, of inflammation or adverse effects also that are being assessed. And this is the way the randomization process works. So you would take a random group of participants. So you're trying to match them. They're trying to match them for age. So you wouldn't want to test your drug in all young people and your placebo in all old people, or the other way around. So in the case of people with the illness, it might be an illness that's only found in older people, in which case you'd also trying to be matched things like ethnicity, sex, gender, health, weight, all stage of the illness, is it early stage, later stage. So you're trying to do your best to make sure that the two groups who get the control or placebo or the current therapy, so the placebo or the current therapy or the new drug are matched. So you've not biased that in any way. So that's the way that's done. And this biasing's done, using a computer software. So then basically nobody knows, neither the doctors nor the patients know which drug they're getting. Are they getting drug or placebo? So that's why it's double blind. So in terms of the trial happening, the physicians or nurses who are delivering the treatment would just give the patient one or other tablet or whatever, and then at the very end of the trial, the code would all be broken, and you'd actually the software would tell you who got what, and then you'd analyze the data. So all of this is trying to avoid the bias in the trial in terms of people with milder disease being given the drug, so then the drug looks better than it actually was, and all that kind of thing. So that's one thing that's done. So placebo, again, a really important concept that can affect anything that we're exposed to. So this would mean that we would get if in a trial you would get a sugar pill or a saline injection or even a sham surgery, if you were trying to find if a new surgery actually worked. And it's meant to prevent researchers and subjects from knowing whether treatment is effective or not. So that people have got this expectation about efficacy, and that can influence the results. And in some circumstances, especially when effective treatments already exist, then the new drug may be given in combination with an existing drug to actually look at whether the new drug actually is a effective add-on or replacement for the existing treatment. So the placebo effect is actually telling us whether there's this expectation of a positive effect that actually leads to a benefit. And I've got a couple of links here, which I'm going to put on Quirk, because this is a short lecture, so we won't have time to look at them now, but I'll put these two links. There's this one and another one. I'll put these into Quirkus. Hopefully you can also access them from the slides, but I'll upload them and you can have a look at them. They're helpful videos to understand the placebo effect. But if you think of the placebo effect of this expectation of a positive result, so you've got expectations, learning, conditioning, verbal cues, emotions, motivation, contextual cues. So all these things kind of psychologically and emotionally are priming us for a certain expectation. And if we put this also in the context of that slide about the spin, so the spin on positive results or an effect of a herb or something on a condition, this is all adding to, whether it's via media or social media or whatever, or that's all adding to this expectation of a positive effect from something, or a negative effect, which we'll also talk about. So this really has a massive impact in clinical trials, and some clinical trials there's been just a huge placebo effect, and it makes it really hard to figure out if the drug's actually working. So it's really remarkable how big this placebo effect can be. So this just gives you a kind of cartoon version of this, but you've got the drug group and the placebo group. So in this case, you've actually, you remember you're measuring outcomes, so you're measuring if the patients actually get better, and it could be with actual measures of inflammation, not just not even just their perception of if they're better, but at concrete measures of, say, a biomarker. And the placebo effect could be significant. So you've actually got a significant benefit of something that doesn't contain the medicine at all. And in this case, it looks like it's maybe, I don't know, maybe a quarter or something of the drug effect, but it can be a lot bigger than in some trials. You've got this huge placebo effect. So this is to do with the physiology of us expecting something to actually help us. And it actually affects our physiology and our biochemistry and inflammation and all sorts of things to actually have an effect. So we mentioned these endogenous opioids, these active opioids in the body, and actually some of the placebo effect is due to the release of these endorphins in our body, natural painkillers. Could also involve cannabinoids, but we'll talk about that later. And so these things are actually produced in our body in response to this placebo, which the patient potentially thinks is the drug. So they think they're getting the drug. And so you get release of endorphin, also potential release of dopamine, and you may actually end up with this pain relieving effect. Again, very important to be aware of when there's kind of what we'd call anecdotal data, so what people are sharing their views on something. Well, this really worked for me. I feel so much better. And in fact, it could really be that someone is feeling better, but is it the actual compound, or is it this expectation of feeling better? So which in effect you could say, well, does it really matter if you're taking something and it's a placebo effect? Well, it matters if the compound might have side effects, and it matters if people are spending lots of money on something that's expensive. So these are all things worth thinking through and understanding as well. And so there's also something called the nocebo effect, which is the anticipation of something negative. And that can be, again, what people tell us, what happens around us, who we are, our past experiences, all actually can lead to this kind of anticipation of a negative effect, and that's actually happening as well. In clinical trials, we tend to have the idea much more prevalent of a placebo effect, because you're being given something that could be a treatment. So there's this idea of a positive effect happening rather than a nocebo effect. But then, so this kind of outlines what's happening with these. So you've got all this effect in our cognitive processing, and the idea of a positive effect versus a negative. And there's the emotional things that are associated with this. For example, hoping for something that could work, trusting in the people who are giving you the medication, like trusting in maybe a doctor or a friend who said this thing works, or whatever. So again, this is kind of interesting in terms of where we get information from and that kind of thing. And then there's these actual real physiological pathways that are impacted by these emotional cues, and actually affecting things like opioids, which are actually natural painkillers, and dopamine, which actually do affect our mood. So without the drug doing anything to those things. And then there's the actual health outcome, which can in fact be pain increase or pain decrease or whatever, depending on the different situations of the context. And this is an interesting one. I think this is just my last slide here. Yes, so this was a recent article of 2022, and interesting, the findings of this review. So we'll talk more about what a systematic review is and what a meta-analysis is in later lectures, because these are important ways that we gather this solid evidence. But in this case, they're saying that it suggests that placebo responses contribute significantly to pain reduction in cannabinoid clinical trials. And they say here that the unusually high media attention surrounding cannabinoid trials with positive results, irrespective of scientific results, may uphold high expectations and shape placebo responses in future trials. So this is really interesting as well, because if there's one positive trial and it's kind of hyped, or you've got the spin, then this is going to have big effects on the placebo effect in future trials, where people are thinking, hey, that worked really well in that last trial. Then there's this heightened sense of anticipation of a positive result. So this, I think, was a really interesting kind of study that's been done in that paper, and specifically on cannabinoid-related, cannabis-based therapies for pain. And maybe we'll look a bit more at this actual, I just found this paper recently, so I'll have another look at it maybe in the upcoming lectures.